The reason why we have these as different labels and different vials is each of those flies is a different genotype.
Okay.
So each of these has a different little new added piece of DNA stably integrated into their genome.
Right.
And we want to keep them separate from one another.
Yeah.
So that we know the genetics of them.
Right.
So that if we ever want to cross them together, we can do that and we know what we've got. And you can see some of these flies look different to one another. So, for example, the flies that we had coming out from here, if you look at the pupil cases, so these pupil cases look like basmati.
Yeah.
These pupil cases look like arborio risotto rice. Yeah?
Yeah.
So these flies, as well as having the piece of DNA that we've put inside them, also carry a slightly different chromosome that has what we call a phenotypic marker on it. It has a visual difference.
Okay.
So you can tell flies that have that particular chromosome with them. So this is a particular marker called tubby. So the larvae are shorter and fatter, and the flies are shorter and fatter than their wild type counterparts.
Okay.
We have various other ones. So that one's, let's get that round the right way. That was in there, that was in there. We have others. So these ones are curly. So they have little, where are they? They have little curly wings rather than straight wings in them. So again, that's just a different, we can see there, little raised curly wings. So we can tell the difference between them. And that helps us when we're tracking flies across generations, because we know through all of those experiments that Mendel did with his peas, and then Morgan did 100 years ago with the flies, that you can track chromosomes across generations. So that theory of heredity that says that there are particular genes on what we now know particular chromosomes, and they move through generations together. So
okay.
That way we know what's on each of these chromosomes. And fruit flies only have 4 chromosome pairs. We have 23 chromosome pairs. So trying to do genetics with humans across generations, not only is it difficult because every generation is 20 to 30 years, but on top of that they have 23 pairs of chromosomes. So tracking everything through that length of time is not
and keeping them in little vials in a laboratory is kind of unethical as well.
There is that. So